Welcome back YouTube, this is my channel of an everyday life of an Aspie. If you're following me and you're into my channel and you landed on my page for whatever reason, I welcome you all with open arms. I'm Aspie and I'm all about creating mental health and wellness and sharing my life stories for speech syndrome and the like, along with tips and tricks along the way. Just to bear in mind with the tips and tricks of what I may give you out, it has worked for me basically, and I thought to share with you my opinions based on how to treat or manage these everyday personal symptoms, or either or. It goes for the same retrospect. If anything that has worked for you doesn't work for me, that's fine. Like I said, give and take. It's all about, respecting one another's differences of opinion. It has also brought to my attention right now, while I've been doing my research and sharing you all these all about series, especially that I'm going to have to do the last one, which is the all about Alzheimer's, maybe 3-4 parts, so it's easier to follow, because obviously with a lot of research I've been doing along with my understanding of it, hopefully some of this will be understandable enough, along with some diagrams and that, to show you step by step what I'm talking about here, so that we can get on with it. So, enough further ado guys, basically this one's obviously all about Alzheimer's, but before I begin this, even though this is the second part based on the genetic side of things, just a disclaimer as always, I'm no medical professional, I'm just your normal everyday Joe Blow, so if you see any clinical signs and symptoms in regards to these health topics I share, regardless being in mental health or general health, especially in these ones that I'm currently doing right now for you, seek professional help or medical attention by seeking second opinion with your doctor, for, like I say, never condone any self-harm. So let's begin this off the part 2, obviously, for this, and I humbly apologise if I'm running off late. So this one's basically a said based on the genetic side of things. So this is hereditary. Scientists know genes are involved in Alzheimer's. There are 2 types of genes that can play a role in affecting whether a person develops a disease: risk genes and the deterministic genes. Alzheimer's genes have been found in both categories, however. Genetic testing. Genetic tests are available for both APOE, E4 and the rare genes that directly cause Alzheimer's. However, health professionals don't currently recommend the routine genetic testing for Alzheimer's disease. Testing for APOE, E4 is sometimes included as a part of research studies only. Risk genes can increase the likelihood of developing a disease, but is not guaranteed it will happen. However, scientists have so far identified several risk genes implicated in Alzheimer's disease. The risk gene with the strongest influence is called apolipoprotein E4, or as I've mentioned before, APOE E4. Scientists estimate that this gene may be a factor in. Just a quick note of the abstract and that about what I'm talking about the apolipoprotein E, or APOE. Basically, this is a major cholesterol carrier that supports lipid transport and injury repair in the brain. APOE polymorphic alleles are the main genetic demonstrations of Alzheimer's disease, so you'll see diagrams floating around somewhere in what I'm discussing here along the way. Obviously, basically for this being the main genetic determinants of Alzheimer's disease, or AD risk, individuals carrying the E4 allele are at increased risk of Alzheimer's disease compared with those carrying the more common E3 allele, whereas the E2 allele decreases the risk. Presence of the APOE E4 allele is also associated with increased risk for cerebral amyloid angiopathy and age-related cognitive decline during normal aging stages. APOE lipoproteins bind to several cell surfaces receptors to deliver the lipids, and also to hydrophobic amyloid AB peptides, or alpha peptides, which is thought to initiate toxic events that leads to the synaptic dysfunction of neurodegeneration in AD. APOE is isoforms differentially regulate alpha-beta aggregation and clearance in the brain, and have distinct functions in regulating brain lipid transport, glucose metabolism, neuronal signaling, neuroinflammation, and mitochondrial function. Obviously, in this review, describe the current knowledge of APOE and the CNS, with a particular emphasis on the clinical and pathological features associated with carriers of different APOE isoforms. Obviously, the alpha-dependent and alpha-independent mechanisms was also discussed, but I'm going to try to make it as short as brief, that will link APOE for status with the AD risk, and consider how to design effective strategies for AD therapy by targeting APOE. As we know, or should know, APOE regulates lipid homeostasis by mediating lipid transport from one tissue cell to another. or another cell type to another. In peripheral tissues, APOE is primarily produced by the liver in macrophages and mediates cholesterol metabolism in an isoform dependent manner. APOE4 is associated with hyperlipidemia and hypercholesterolemia, which leads to atherosclerosis, coronary heart disease, and stroke. 11-12, or in the CNS I should say, APOE is mainly produced by astrocytes and transports cholesterol to neurons via APOE receptors, which are members of the low-density lipoprotein receptor LDLR family. APOE is composed of 299 amino acids and has a molecular mass of 34 kDa. Differences between the three APOE isoforms are limited to amino acids 112 and 158. Where either cysteine or arginine is present, APOE2, size 112, size 158, APOE3, size 112, ARG 158, APOE4, ARG 112 and ARG 158. The single amino acid differences at these two dispositions affect the structure of the APOE isoforms and influence the ability to bind lipids receptors and alpha-beta 13 to 15. Human and animal studies clearly indicate that APOE isoforms differentially affect alpha aggregation and clearance. Several alpha-independent functions are also associated with APOE. So, in response to this, the APOE and alpha disposition, just to illustrate, APOE has an important role in alpha-beta metabolism. Studies show that APOE genotypes strongly affect disposition of alpha-beta to form senile plaques and cause cerebral amyloid angiopathy, CAA, two major hallmarks of amyloid pathology in AD brains. Immunohistological evidence demonstrates that APOE is co-deposited in senile plaques in the brains of AD patients. The alpha-beta deposition in the form of senile plaques is more abundant in APOE4 carriers compared with non-carriers. This difference was most evident among individuals between the ages of 50 to 59 years. 40.7% of APOE4 carriers had senile plaques compared with 8.2% of the non-carriers. In individuals with positive Pittsburgh compound, images which will locate fibril aggregates of alpha-beta was more common than those in the negative scans. 65% versus 22% in patients with AD2.9. Fibrillar alpha deposition is often detected in the brains of elderly cognitively normal individuals in a manner that depends on the presence of APOE4, although such an association is weaker than in patients with AD. In addition, APOE4 carriers has lower cerebrospinal fluid or CSF, alpha-beta 42 levels and higher PIB positive imaging, which reflect the presence of cerebral amyloid deposition and serve as potential biomarkers for AD. Cognitive normal APOE4 carriers exhibits PIB positive imaging about 56 years of age compared with about 76 years of age in non-carriers. The difference suggests that APOE4 probably increases the risk of AD by initiating and accelerating alpha-beta accumulation, aggregation, and deposition in the brain. Although APOE2 reduces the risk of dementia in individuals older than 90 years, both the E2 and E4 alleles of APOE increase amyloid burden compared with APOE3, suggesting that the protective effects of APOE2 means AD might not be associated with the alpha-beta deposition. APOE4 also shows an association with CAA and CAA-regulated hemorrhages. CAA refers to the pathological condition in which amyloid spreads and deposits through the cerebral blood vessels walls and is frequently detected in AD. Interestingly enough, although APOE2 is protective against AD, it is a risk factor from CAA-related hemorrhage independently of AD, possibly by predisposing vessels to vasculopathic complications of CAA. So this quickly ends the part about the deposition and everything else. Sit tight and I'll... Just a quick note before I continue, for some of these abbreviated terms that I was mentioning, obviously you may be wondering what CAA is. So CAA stands for, in the medical terminology, cerebral amyloid angiopathy, which is also known as congophilic angiopathy, which is a form of angiopathy in which amyloid deposits form in the walls of the blood vessels of the central nervous system, basically. So the congophilic is used because the presence of the abnormal aggregations of the amyloids can be demonstrated by the microscopic examination of the brain tissues that is used. However, and this term obviously also, after the application of a special stain called Congo red, when you do this, the amyloid material is only found in the brain, and as such the disease is not related to any forms of the amyloidosis that you may hear about or whatever else. So this quickly ends the Here on the top left of the screen here illustrated is the biology of APOE, gain and losses, to clearly illustrate what it's all about, to gain an understanding on what I'm talking about. Sit tight and I'll bring you more information soon. 5% of Alzheimer's cause cases. APOE4 is one of the three common forms of APOE gene. The others are APOE2 and APOE3. Everyone inherits a copy of some form of these genes from each parent. Those who inherit APOE4 from one parent has an increased risk of Alzheimer's. Those who inherit this APOE4 from both parents have an even higher risk, but there's no such certainty here. Scientists are not yet certain how APOE4 increases this risk. In addition to raising risk, basically, the APOE4 may tend to make Alzheimer's symptoms appear at a younger age than usual. Deterministic genes directly cause a disease, guaranteeing that anyone who inherits them will develop the disorder. Scientists have discovered variations that directly causes Alzheimer's disease in the gene's coding of the three proteins: amyloid precursor protein APP, presenilin 1 PS1, and presenilin 2 PS2. When Alzheimer's disease is caused by these detrimental variations, it's called autosomal dominant Alzheimer's disease, or ADAD, or familial Alzheimer's disease. And many family members in multiple generations are affected. Symptoms nearly always develop before the age of 60, however, and may appear as early as 30s or 40s. Detrimental Alzheimer's variations have been found in only a few hundred extended families worldwide. True familial Alzheimer's accounts for less than 5% of cases. Genetic testing for Alzheimer's disease risk assessment. Alzheimer's disease has a strong hereditary component, which researchers intend to investigate along to detect a more effective form of treatment of this common form of dementia. As I said before, this is a form of dementia. Scientists have recently discovered 11 new genes of degenerative disease, doubling the number of potential strategies that can design drugs designed to stop, hopefully, memory loss. The new genes associated with Alzheimer's. An international team of researchers from the Institute of Genomics at the University of Medicine in Miami, USA, analysed the genetic data collected from more than 25,500 Alzheimer's patients and more than 49,000 individuals from 15 countries. This research has led to the discovery of these 11 genes that are associated with predisposition to the development of to define Alzheimer's disease, in addition to those already identified in the middle keywords. Scientists have identified 13 genes that are still needed to be further investigated, which may involve in triggering the degenerative neurological disease. The findings provide a broader overview of the genetic factors that contribute to onset of Alzheimer's and a better understanding of the disease seen from new angles and perspectives from all these researchers. These new perceptions include the immune system, which could be a key factor in the onset of neurodegenerative diseases, which includes sclerosis or Parkinson's syndrome. Many new genes believed with certainty that would be involved in the development of Alzheimer's disease it has already been known biological processes of the disease. However, moreover, one of the new genes was confirmed as part of the genome that play a crucial role in the immune and inflammatory response. The discovery suggests that disease shows an abnormal accumulation of proteins in the brain. Alzheimer's disease, Parkinson's disease, and MS may have a common mechanism which once was known to that can help to develop a more effective treatment. Although there is a real form of Alzheimer's disease that occurs before the age of 65 for most patients, the most common types of the syndrome begins after this stage. The most common gene associated with Alzheimer's is called apolipoprotein E. This protein has three main forms. One, APOE2 protein, which is the most rare. Two, APOE4, most common protein that increases the risk of the onset for most patients, and 3 is APOE3, which is the most common protein. Thus, people who inherit one of these APOE genes from the mother and one from the father are born with 2 copies of the APOE gene. Presence in the DNA, at least one gene, APOE4, significantly increase the risk of developing Alzheimer's disease. If 2 genes are inherited, APOE4 disease risk is even higher. Important to remember though that not all persons that carries this APOE4 gene develops Alzheimer's disease, even if syndrome occurs in most cases among patients with APOE4. That is also only considered a risk factor and not the actual cause of Alzheimer's disease, participating along with environmental factors to this disease. As research on the genetics of Alzheimer's disease progresses, scientists have discovered new links between diseases occurs after the age of 65 and many other genes in the genetic codes. The most important of them are 1. SORL1. Some variations of this gene presents in itself in the chromosome 11 appear to be associated with Alzheimer's disease. CLU. This gene is designed to control the release of substance called amyloid beta in the brain. Researchers believe and support the theory that the imbalance in the production and release of amyloid beta can trigger Alzheimer's syndrome in old age. CR1, a deficiency in protein produced by this gene may contribute to chronic inflammation of the brain. Inflammation is another possible factor involved in the development of Alzheimer's disease. PICALM, this gene in question is correlated to the processes by which the brain nerve cells of the neurons, how they communicate with each other. Corresponding transmission of information between neurons is important for the normal brain function and maintain long-term memory. TREM2. This newly identified gene is involved in regulating brain response against inflammation. Rare TREM2, however, gene variations are associated with increased risk of developing Alzheimer's disease. As of APOE gene, the above are considered risk factors, not direct causes of the degenerative neurological disorders. In other words, to explain further, the presence of several variations of these genes, one or more, may increase susceptibility to Alzheimer's disease, but not a certainty about the occurrence of the syndrome. Most medical experts, however, do not recommend genetic testing for late-onset Alzheimer's disease after 65 years of age, but they may be necessary in the event of symptoms syndrome before the early ages of the 65. If the APOE gene, for example, the simple determination of presence in this genetic code doesn't say whether the disease will develop or not. Moreover, some people who have the gene APOE4 in DNA develop Alzheimer's disease, as a significant number of people with this gene doesn't get sick of this form of dementia. For this reason, doctors discourage genetic testing for APOE genotypes because the results are difficult to interpret to know for certain which is which. So this quickly ends the genetic hereditary stuff about the Alzheimer's. Give me the like for thumbs up for support, comment below if I missed out anything. Feel free to follow me to my social media sites as it's linked below. Feel free to subscribe to my channel if you haven't done so. Feel free to switch on the notification bell to keep up to date of all the new videos that is coming to effect as of now and till whatever time. Feel free to share these videos around to family and friends so that we can learn from each other regardless, or use it as a resource tool, whatever it may be. So enough with the do, thanks for your support, thanks for watching, do what you love what you do. Until next time, Aspie signing out, I'll see you again soon. Ciao. Just a quick brief summary of genetic testing so that you guys understand further, even though it was a bit more in depth to what I've illustrated to you all. So as we know, genes play a role in the development of many types of dementia, although the extent of this role varies between dementia types. A person with a parent or sibling with a dementia such as Alzheimer's disease has on average a higher risk of developing the same condition themselves, although other factors such as lifestyle can also play a part also. At present, the relationship between genetics and dementia isn't fully understood. We don't know that a mutation or the change in one of the small number of genes can cause someone to develop frontotemporal dementia or any rarer inherited forms of Alzheimer's disease. These conditions most often start in people under the age of 65. Predictive genetic testing is where the genes of someone who is related by birth to a person with diagnosed with dementia are tested. The genetic test aims to see whether the relative has inherited the same mutation and so will go on to develop on the stages of dementia. At present, such predictive genetic testing is also only possible for inherited Alzheimer's disease, in which very rare mutations in 3 genes are implicated, and frontotemporal dementia, which has known mutations in at least 6 genes. The most common form of Alzheimer's disease affects about 520,000 people in the UK. Most often starts in people over the age of 65. There are no approved predictive genetic tests for this form of the condition. In the vast majority of people, any genetic risk of Alzheimer's disease, increased or decreased, is linked not to rare mutations but to variations in a large number of susceptibility genes that they may have inherited. The most common risk gene is called the APOE, as mentioned earlier. This gene has a number of variants, and a variant called E4 has been shown to increase risk of developing Alzheimer's disease. However, risk genes such as APOE have a more subtle influence on someone's chances of developing Alzheimer's disease than the genes that are used in tests for the inherited forms of dementia. Genetic testing can be difficult emotionally, not providing any conclusive results, and can cause practical difficulties. Before considering genetic tests, however, it is vital that proper genetic counselling is provided to ensure it is the correct decision for the individual involved. It is also vital that someone does have testing, they do receive full genetic counselling about what the results make. No counselling is currently provided with the commercially available APOE test. While this quickly ends the summary of genetic testing, sit tight and I'll bring you back more.